Okay, before we begin, are there any questions? Oops, that's the wrong Zoom. My computer's going to be slow because it's still processing the, gosh, how do I get Zoom now? Oh, there it is. It's still processing the lecture Zoom file, making it an MP4 file. Let me share my screen. Ah, there's the lecture. It's only 69%, so I'll be doing this for a while. All right, today is February 26th, and today will be our last lab. All other days, we will only really be working on the unknown project. Let me remind you, your unknown project is due Saturday, March 9th. The unknown notes pages will close on Friday, March 8th. So the last day to request a test would be Friday, March 8th. And if you request a test on Friday, it must be a test that you will incubate for a day or less. So for most of the tests, you'd have to request a test on Thursday, March 7th, because most of them will take 2 days to incubate. Any question about any of that? That is only a week from this Saturday. So your unknown project is going to be finished pretty soon. Hopefully you guys are well along on getting it finished. Those of you who haven't started, you're probably not going to have time to finish the project now. All right, any questions on what we're doing? Lab 9, identification of gram-negative organisms. If not, let me go ahead. I don't like closing this, but I'm going to because my computer is going to be slow until that finalizes. So that looks a little big. Let's go there. Lab 9, identification of gram-negative bacilli. There is some pre-reading, not very much, to do for Lab 9, page 131 to 132, so one page on biochemical tests and identification, and then about 4 pages on methods of classifying and identifying microorganisms. There are some video clips in today's lab. Upon successful completion of this lab, you should be able to, 1, interpret the results for the following biochemical tests for the IMVIC reaction. IMVIC is an acronym for indole, methyl red, Voges per square, and citrate. So this is actually 4 different tests. Indole test, the methyl red test, the Voges per square test, or the citrate test. And I may be butchering this name because I'm not Russian. So I'll just, from here on, I'll call it the VP test. We'll also talk a little bit about the carbohydrate fermentation, the motility test, and the hydrogen sulfide production test. We should also be able to explain how to interpret the TSI test, and that stands for triple sugar iron, the SEM deep test. Oh, good, that just finalized. Let me save that down. And this is an acronym for sulfur indole motility test, and be able to explain what the growth on McConkey's means, and also the growth on hectone enteric agar, or HE agar plates mean. And then 3, use biochemical analysis to solve case studies involving gram-negative bacterial infections. Any questions about the objectives? If not, let me put this down here. Maybe I put it over there. All right, members of the family Enterobacteriaceae are all small gram-negative, non-spore forming bacilli. This is one of the largest families of bacteria. They are all facultative anaerobes. They have 53 genera in this family. 24 of these genera have been known to cause human infections. The members of this family are all glucose fermenting, oxidase negative organisms. And here we've seen showing some of the genera that are actually normal microbiota of the intestinal tract of many animals, aka coliforms, like Escherichia, you know, is in your intestines. Enterobacter is in your intestines. Salmonella and Shigella are not in your intestines, but they are in the intestines normally of birds. And that is one reason why you should be very careful when you're handling, well, chicken or any other bird, because they have Salmonella and Shigella in their intestines. And when we get these two organisms, these two species, they can cause illness in humans. Anyways, the Enterobacteriaceae, especially the ones shown above, are all opportunistic pathogens in that they can cause urinary tract wound infections. They can also cause pneumonia and septicemia. So if they're growing where they should be, they are part of our not nosocomial. They're part of our microbiota if E. coli is growing where it should be. But if it starts growing elsewhere, it will cause an infection. So common genera responsible for causing opportunistic infections in human include Escherichia, Enterobacter, Klebsiella, Citrobacter, and Serratia. Several genera are also involved in gastroenteritis, meaning they're frank human pathogens. And that includes things like Salmonella, Shigella, as well as E. coli, Serovar 0157H7. This variant of E. coli is a human pathogen, and these antigens cause illness in humans. Any question about that? So E. coli, when it's in your gut, does not normally cause humans any problems. In fact, we have E. coli in our gut, and it helps us digest our food. However, this strain of E. coli that has the antigens 0157 and H7 is a frank human pathogen. There is another family of gram-negative bacteria that is very similar to the Enterobacteriaceae, and that they are all gram-negative bacilli, and they're short gram-negative bacilli also. There are some differences between the Pseudomonaceae family and the Enterobacteriaceae family, in that Pseudomonas are all non-glucose fermenting. They're all oxidase positive. And if you remember, Enterobacteriaceae all ferment the sugar glucose, and they're all oxidase negative. The Pseudomonas family can be opportunistic pathogens, particularly Pseudomonas aeruginosa, which is often involved in infections involving immunosuppressed individuals, burn patients, and trauma patients. And these infections are often called healthcare acquired infections, which we previously called nosocomial infections. Any question about any of that? So Pseudomonas aeruginosa is a frank human pathogen, but it's lousy at getting established in a healthy individual. You need a break in either your skin or your mucous membranes before Pseudomonas aeruginosa can usually cause an infection. And obviously burn and trauma patients have breaks in their mucous membrane and their skin. And then immunosuppressed individuals are just lousy at throwing off Pseudomonas aeruginosa. It's important for microbiologists to be able to identify and perform tests on gram-negative bacilli that are sent to the lab for culture as a clinical specimen. In this lab, we're going to talk about some of the biochemical tests that are used to identify gram-negative bacilli. And here you're looking at a whole slew of different tests. We're not going to talk about all of them that can be ran to determine the identification of the species, especially of gram-negative bacteria. And what you do here is you have this little device here, and it has a little pin, and you stick the bacteria on the pin, and you insert the pin inside the tube, and then you're running 12 tests at one time. We'll talk about the citrate test, the lactose fermentation test. We'll talk about hydrogen sulfide production, and then glucose fermentation also. But all the other tests we're not going to talk about. Well, we'll talk about the VP test. Sorry, didn't see that there. All right, the first test we're going to talk about is the indole test. Some organisms produce the enzyme tryptophanase that converts the amino acid tryptophan into the products of indole and pyruvic acid. You have overnight incubation, and then you add Kovacs reagent to the tube. Kovacs reagent reacts with indole to produce a red color at the surface. E. coli tests positive for indole, and Klebsiella pneumoniae does not produce indole. So what you do is you have a broth that has the amino acid tryptophan in it, and that amino acid could simply be in protein. So it doesn't have to have the amino acid tryptophan directly added to the media. It could just have the source of protein providing the amino acid tryptophan. In the presence of tryptophanase, you produce indole, which can react with Kovacs reagent to create a cherry red color. And as you notice, that's not very cherry red. I think that's partly the photograph. That kind of looks like more of a purple color, but that's indole positive, and normally it's a cherry red color. And then the negative will remain a yellowish color. Any question about the indole test? If not, we're going to talk about the methyl red and the Voges-Perskier test. The MR test and the Voges-Perskier, I'm going to henceforth call it the VP test, are tests used usually together, meaning what you do is you inoculate one tube of MR VP media, and then you split the media in two. One tube you use for the MR test, the other tube you use for the VP test. Both tests, the MR and the VP test, are used to detect the end products of glycolysis. The MR test checks for the presence of acid products. The VP test checks for the products of glycolysis that are neutral products. So the MR test is looking for the production of acid. The VP test is looking for the production of a neutral byproduct, which is shown down below. Nope, I don't even have it here. So both tests are looking at glycolysis. What you do is, like I said, you inoculate one tube, incubate it, and then split that tube into two tubes. One portion used for the MR test, one portion used for the VP test. In the MR portion, you run the MR test by simply adding methyl red to the MR tube. And if acid products are made, the pH indicator methyl red will remain red in color, and that will be positive for the MR test. If the methyl red, when you add it, changes color to yellow, then that would be methyl red or MR negative. Any question about any of that? So there is an MR positive result, there is an MR negative result. So when you added methyl red here, it quickly changed color to yellow. And this remains red. So methyl red positive. All right, for the other half of the tube, you then use it for the VP test. You have to add Barrett's reagent A and Barrett's reagent B to the tube, as well as shake it to add oxygen to the tube, and then let it sit for 30 to 60 minutes. We're looking at the presence of acetone, and if acetone is in the tube, it'll react with the Barrett's A and the Barrett's B and the oxygen to produce a red product. And at usually the top of the tube, there will be red in the VP positive tube, and the VP negative tube will not turn red. It'll remain yellow in color. Any question about that? What's happening is acetone is just reacting with the reagents to become red in color, but this doesn't happen immediately. Why it needs to incubate 30 to 60 seconds, or not seconds, minutes. Any question about the MR or the VP test? Now, what's easy in all of these three tests is the MR test, the VP test, and the indole test all turn red if they are positive. And they are yellowish if they are negative. All right, let's move on to the fourth test of the Einvik test, and that's the citrate utilization test. This test is looking to see if the bacteria can metabolize citrate, the sugar, and if it can metabolize citrate, it will grow on the tube and change the color of the tube from greenish to bluish. Citrate, if it's metabolized, will make a basic end product, which will change the pH, and this pH indicator will switch from green to blue if citrate is metabolized. And you should see a growth on the tube. This tube's not showing you any growth, but you can assume there's growth there because it's blue. If the bacteria cannot utilize citrate, it'll have no carbon source in the tube because citrate is the only carbon source. And so the bacteria will not grow, and it should not produce bases because it's not metabolizing, and the tube will remain green. Any question about any of that? All right. So there we're looking at citrate. It's in the Krebs cycle. Obviously, a bacteria that can perform aerobic respiration can metabolize citrate. But not all aerobes can metabolize citrate. But if it can, it'll be citrate positive. And if it doesn't have the Krebs cycle in it, then it cannot use citrate, metabolize it. All right, any questions about the citrate utilization test? If not, let's talk about the PR carbohydrate metabolism test. This tube has one sugar in it, which can be fermented by the bacteria. And if the bacteria ferments that one sugar, it will produce acid byproducts. The acid byproduct will change the pH indicator to the color yellow. And so if you see yellow color, you know that acid has been made, and that means that the sugar in the tube has been fermented. Now we call this the carbohydrate metabolism test because there can be different sugars in the tube, but we only have one sugar. And so you could call it the glucose metabolism test or the lactose metabolism test or the sucrose metabolism test. But to talk about them all together, we're just going to call it the carbohydrate metabolism test. Looking at the fermentation of the one sugar that's in the tube. The tube also has an inverted Durham tube. That's that little tube inside. It's filled with media. If the fermentation can produce a gas, you'll see a bubble that'll be made in the tube. And you'll only get the bubble if fermentation occurs. Any question about any of that? All right. So that's carbohydrate fermentation test. Let's move on to the motility test. The motility test is a semi-solid media, meaning it's very thin, sort of like Jello, and you stab inoculate it. If the bacteria only grow in the stab, that means the bacteria are non-motile. If, on the other hand, the bacteria flare around the stab, and I'm trying to show you a flare, let me draw it a little better. Well, that didn't work. Why didn't they bring up paint? There we go. So we have a tube like this. We stab inoculate it. If you see bacteria growing out of the stab in flares, and they could be different on the other side or they could be the same. I'm going to make this one a little different than that one there. The flare means that it has motility, but it's not great motility. If the bacteria has great motility, you won't see the flares. You won't see the stab. It'll be a cloudy color like that. And that's because the bacteria have swam everywhere in the media. And so if it's cloudy everywhere, or if the bacteria flaring out of the stab, then it's motile. If the bacteria only grow within the stab, it's a non-motile bacteria. Non-modal means it is lacking a flagella. All right, any question about the motility test? Now let's talk about the hydrogen sulfide production test. Hydrogen sulfide production test is looking at the production of hydrogen sulfide. You can run this test in different tests, such as the triple sugar iron test, the sulfide indole motility test called the SEMS test, and on the hectone enteric auger. All of them will look for the presence of a black precipitate. And if you see the black precipitate in any of these tests, that'll mean hydrogen sulfide has been produced. Citrobacter, Salmonella, and Proteus are hydrogen sulfide positive. This is a hydrogen sulfide positive, and that's a hydrogen sulfide negative bacteria. Any question about hydrogen sulfide? All right, this is showing you hydrogen sulfide in the SEMS test turning black. You could get it in the triple sugar iron test, or the TSI test right here. And let me see if I got a picture of that. Right there, there's black precipitate in TSI. And right here is a black precipitate on hectone enteric auger plate. All of them indicating the production of hydrogen sulfide. All right, let's talk about the triple sugar iron slant. This test runs several tests all at once. The tube has in it 0.1% glucose, 1% lactose, 1% sucrose. And it has sodium thiosulfate and ferrous sulfate, which allow the production of hydrogen sulfide. If the bacteria grow on the slant, meaning we inoculate the slant to get bacteria to grow on it, and that will be looking at aerobic growth. And if we inoculate the slant, we'll also be looking at anaerobic growth on TSI. You then look to see the color of the media in both the slant and in the butt, and they can differ, as well as the black precipitate. What's the third test you're looking at in that one? Oh, the production of gas. You can also see the production of gas. So this test is rather complicated. In this tube, it's negative. There's no change in the color of the slant, no change in the color of the butt. And so we have that K over K. K means no change. In this tube, we have K in the slant, and in the butt, it's black. So we have the production of hydrogen sulfide gas, because it's black. If ever we have the production of hydrogen sulfide, you can know that fermentation occurred. So fermentation is in the butt. And so we're going to abbreviate that as K over A, meaning no change. You can also put that negative in the slant, and then A in the butt, because the butt would be yellow if we could see the yellow, but we can't see the yellow because it's covered in black. But if ever you see black, you can assume it is yellow, because only when fermentation occurs does hydrogen sulfide production occur. And so this would be what this tube would look like if you remove the hydrogen sulfide production. So in this tube here, we have K over A, and like I said, you can have that negative over A, and that means no change in the slant, acid in the butt, no hydrogen sulfide in the butt because it's not black. I'll talk about the glucose and the other sugars being fermented in a minute. In this tube, we have A over A because it's acid or yellow in the slant and in the butt. We see the production of gas here, and that may appear to be a broken auger or a slight bubble right there. This one has a very large bubble at the bottom. So there is gas produced in this one. Because the color is yellow in both the slant and in the butt, we're going to say that glucose is fermented, plus either lactose or sucrose, or both are fermented. And that's because if only glucose is fermented, glucose will be used up in the slant because the bacteria grow very rapidly, aerobically, and the glucose will be used up. So the color changes from yellow to red because once the glucose is used up, the proteins will be metabolized. When proteins are metabolized, they will produce, you pull off the ammonia, and that'll be excreted by the cell, usually by as urea or something, uric acid, or maybe just ammonia. And that will be basic, and that'll change the color from yellow to red. So if we were to look at this color early on, we would see that the slant was initially yellow, but later on, all of the glucose is digested because it's at low concentration, only 0.1%. And so the growth aerobically will use up all the glucose and then start using the proteins, reverting the color from yellow to red. The slant, on the other hand, anaerobic growth, the bacteria grow slower, they digest the glucose slower, and so the yellow remains yellow in the bud. So if ever you see it like this, you know glucose is fermented and only glucose. Sucrose and lactose is not fermented because they're at higher concentration. And if the slant and the butt are both yellow, then you know, yes, glucose is fermented because it's the base sugar, but at least one or both of the other sugars are fermented because it's yellow in the slant as well. Any questions about the TSI test? If not, let's talk about the sulfur indole motility test. This test looks for the production of hydrogen sulfide gas, and it'll be black if that occurs. It looks for the motility, that's what the M stands for, and it looks for the production of indole. What you do is you stab inoculate the tube, let it incubate, and then when you bring it out, you add Kovacs reagent to the top. If Kovacs reagent turns red, right there, right there, right there, and right there, then you know indole has been produced in tubes 1, 2, 3, and 4. If the tube does not turn red and remains yellowish, then indole is not produced, and so these are indole negative. If the tube turns black, especially in the bottom, it will be hydrogen sulfide will be produced. Tube 1 is the only one showing that. And then you look at the stab mark. If you see the stab, that means the bacteria is only growing in the stab. I think these two are both negative for motility because the bacteria is only growing in the stab. This tube is clearly positive for motility because the growth is outside the stab. This one here, you can't see it easily, but the reason why it's black everywhere is because the bacteria are motile. And so that the stab mark is actually everywhere. Any questions about any of that? All right, McConkey's and Hectone enteric agar. Look at the growth on the bacteria. If you get growth, then you know the bacteria are gram negative because both of these media select for gram negative and they select against gram-positive. So gram-positive usually do not grow on both plates. And then you can look at lactose fermentation. If the bacteria do not ferment lactose, they will be their normal color, which in this case is off-white or white, but it could be whatever color the bacteria is. On McConkeys, if the bacteria and the media around the bacteria turn purple or pinkish, then the bacteria are fermenting the sugar lactose, creating acid products, changing the color to this color. The HE plate, if the bacteria ferment the sugar lactose, the bacteria will have a salmon color. That's a yellow-orange color. And so this bacteria can ferment the sugar lactose. This one cannot, not salmon yellow colored. And like I said, you can look at hectone and enteric, and if it's black, the colonies are black, that means it's producing hydrogen sulfide gas. All right. While you're not going to perform any of these biochemical tests, because everything's going to be done online, you will be held responsible for knowing how to perform and interpret these tests. We have a video for you to watch on the carbohydrate fermentation test, another one on the SIMS, sulfur indole motility test, another video on the MR or VP test, and then a video on the TSI triple sugar iron test, and lastly, a video on the McConkey's auger test. So watch these videos for this lab. We also have a website, microBuzz. I don't know why that's so small there. Let me look at the worksheet, see if it's that small. Oh, I don't have it on here. Anyways, click on the microBuzz worksheet. You can use the microBuzz website to get some of the information for filling in the table for question number one of the worksheet. And, oh, come on. When you open this up, let me see if I can open that. You only click on one of the links. There will be other links on the left there. See how slow my page is? I think it's the differential test you're clicking on. Click on that. And then these are the tests some of the tests we'll be looking at. You can get the results from some of the tests here. Like there's the citrate test. So use that information to fill in the worksheet. The first has been filled in for you for the positive result of the oxidase test. The cells or the spot turn blue, and the negative result would be obviously they're not blue. And so fill that in for indole, methyl red, Vogel's Prosphere, citrate test, and all the other tests. And then answer the questions for 2. What reagent must be added to detect the presence of indole? 2B, methyl red is a pH indicator that remains what color under acidic conditions. 2C, the what test uses Barrett's A and Barrett's B reagents to detect the presence of acetone. 2D, Simmons citrate uses the pH indicator. Give me the name of the pH indicator, which turns blue under acidic or basic conditions. You need to either bold or write or underline the answer here. So you have 2 questions in 2D, this one here, and then you must need to give me whether it's acidic or basic. You can either bold the answer or underline it or just write it over here. Okay. 2E is the same way. What is the pH indicator used to detect PR carbohydrate fermentation? And then and it is yellow under acidic or basic conditions. F, organisms that possess this structure test positive for motility. G, 3 organisms that produce whole hydrogen sulfide are 1, 2, 3. You can give me either species or families there. If an organism ferments glucose, but not lactose or sucrose, the TSI slant will be what? For the slant, what color will it be? And then what color for the butt right there. And then state the 3 tests which are occurring in the SEMS test. The SEMS tube has 3 tests, 1, 2, 3. And then 2JA, MAC and HE plates are both what? In that they inhibit the growth of gram-positive or gram-negative bacteria. And so you need to write or bold or underline one of these. And that's 2B or 2JB or 2J2, I guess 2J1, 2J2, and then 2J3. They're also what? In that they separate various bacteria based on visual differences from sugar fermentation. Any question about any of that? All right, and then 3, complete the case studies. Use the key shown here, meaning if it's Pseudomonas, it'll be oxidase positive, glucose will be negative, lactose fermentation will be negative, TSI, triple sugar iron, will be K over K. Hydrogen sulfide, we don't have the data for, but citrate will be positive. So use this key only for the bacteria presenting in this lab. Do not use another key, only use this key for this lab. Do not use this key for your unknown project because it won't work. And then this is a table for filling in the results for case study 1, case study 2, whatever. So if oxidase is discussed in case study 1, put the results there. If glucose fermentation is discussed in case study 1, put the results here. So fill in your table here and then figure out what bacteria it is using the key and this table here. And then put in the case study unknown, the species. You need to name the species for case study 1 here. Any question about any of that? Let me see where I've got the case studies. I might have it at the back here. Yeah, I do. It's on the back here. So for case study 1, you can read it, but there's the different tests they're running on case study 1. And there's some tests on day 2, case study 1. You have to scroll down to get what this is. Sorry, this broke on the page, but this is the SEMS tube. So that test, the MR test, the VP test, the citrate test, and then the triple sugar iron test. And then move down to case study 2. Mikey talks his mom into buying a pet turtle. And then Mikey becomes sick. And then there's some few tests they run. That's a colony of interest whenever they have a arrow at it. And then here's the test 2 of the SIM test, the MR test, the VP test, citrate test, the TSI test. So go ahead and read through the case studies and fill in the table and then answer what the species is causing the infection. Any question on this lab? If not, I will be here until about 8 o'clock or the last student logs out to help you with the lab. Go ahead and get started on the lab. Close some of these down. I'm going to step away from my desk for a few minutes, but I promise to be back in no more than 2 minutes. All right?
No rush, you're good.
Okay, good.
So with the unknown project, I was reading more and I guess I was confused, so hopefully I can make up for lost time. Um, so in the unknown notes, I'm supposed to like submit any test requests that I have. So like, am I supposed to like ask you to perform a test in a sense, and then you would like email me the result of that test?
In your unknown notes, you would ask for a test, and then I would incubate it for you. And after the incubation period, I will give you the results in your unknown notes pages. Everything from now on will be done in the unknown notes pages.
Okay.
Okay?
Okay, thank you. Yeah, I I think it might be too late now, but I got confused.
You could be very lucky, so I would try. Obviously, if you're not lucky, you won't finish the project in time.
Sorry.
But if you're lucky, you could. Just
I
depends
think
on,
I'm lucky.
yeah, just depends on how much luck you get, which test you ask, and how lucky you are.
Okay, I see.
And
Yeah,
you do
because
have
I
a
mean
little time. You do have a week or so, over a week actually.
yeah, I have a decent grade in this class. I'm actually not trying to sound bad. I'm actually surprised though, because this is kind of a tough class. So hopefully I can make up for it. But anyway, thank you for your help. I'm gonna get off here.
Okay, I think you're the last one, just to warn you.
Yeah, I know. Thank
Okay.
you.
All right.
Have a good night. Bye.
All right. Good night.